I want to start by thanking Marco and Lisa for organizing. This has really been a mind-expanding conference for me. I'm a biologist by training. I am not a philosopher. I've never taken a philosophy course, although I feel like I've taken one by the end of today. Who knows by tomorrow? As I said, I'm a biologist. I'm going to try to make some connections between what I do as an experimental biologist and some philosophical concepts.
I thought, I know that it's difficult for biologists, but you can stay.
Yes, it's really painful for me not to be moving, but okay, so here's how my talk will go. I'm going to talk very briefly about my journey as a scientist, and then I'm going to introduce a concept known as strong inference, and then I'm going to sort of try to unpack certain parts of the scientific method that I think don't get a lot of attention that should, and then hopefully take some of the lessons that we encounter along the way and pull some of those out to talk about how to teach scientific method better. So, spoiler alert, you know, my answer to this question is yes, and I'm going to explain that more in a minute. Basically, what happened to me was that I labored under some pretty bad misconceptions about how science worked. And it wasn't until some philosophers set me straight that I really started doing what I would consider good science. So what was my scientific journey? Like many of us in this room, I was super interested in science as a kid. I majored in biology. I was enamored with biology as a field. With biological knowledge, I was just amazed by the things that biologists had figured out. I didn't know anything about research, so I thought the best way to stay in biology was to become a teacher. So I trained as a high school teacher. I got a master's in teaching. I taught high school science for a couple of years. And after a couple of years of teaching science and all the products of science, I eventually, got curious about like, how did all of this knowledge get created? And I knew nothing about that. And so I decided to do a master's in animal physiology. And that's when I sort of got sucked into research in biology. And I eventually did a PhD in animal biomechanics and postdoc, and eventually became a faculty member. But in this long journey of being super interested in science, it really wasn't until I was an early PhD student that I would say I figured out how science worked. And that's kind of embarrassing for me to show this slide, that it took me that long to really appreciate how science works. I don't think I'm the only person who had this kind of journey, and so I would love to share some of the things that I learned along the way and with my students and prevent them from waiting so long. So most of us probably had something like this in high school where we were introduced to the scientific method. Starts with observation, maybe a question, hypotheses, prediction, experiment, and then analysis at the end. And this is a pretty good algorithm for how science works most of the time. What happened to me over time was that as my biology training got more and more sophisticated, I was spending more and more time obsessing about experimental method. And what I think got lost was, thinking about these parts of the scientific method, which I think are absolutely crucial. And I'm going to talk more about those later in this talk. And then, statistics becomes a really important thing to know about in biology. So you spend a lot of time thinking about how to analyze the data. Okay, so I've identified a few main misconceptions that were problematic for me as a developing scientist. The first one is something that's actually taught in high school. Maybe you were taught this as a student at some point. And it's this idea that hypotheses are educated guesses about how an experiment will turn out. Okay. That concept, I think, has done more damage to the teaching of scientific method than any other. And hopefully I'll explain why that is. Another thing that was really not useful to me was this idea that, I'm not sure if it was ever taught explicitly, but it's something that I really picked up on interacting with biologists. And that is that, you know, the best questions are the ones that you can express as, what is the effect of this variable on that variable? And, you know, those kinds of questions sound very scientific. But it turns out that if you're only doing those kinds of questions, then you're missing a lot. And I'll flesh that out more later. This is also not something that was explicitly taught to me, but if those first two things are correct, then this is kind of a bad implication of those first two things, that science is just sort of collecting observations, doing clever experiments, analyzing, using clever statistics. And if we collect enough facts, those facts will sort of self-assemble into real scientific knowledge. And we all know that this is a fallacy. So how did I get set straight? Well, when I was a young PhD student, my first year, this paper landed on my desk. I don't know who gave it to me, but it was like a bolt of lightning for me. And it just caused a phase change in how I thought about science. Raise your hand if you've ever heard about this paper by John Platt called Strong Inference. Okay, there's a couple of you, not too many. So let me explain a little bit about this paper. So John Platt was a biophysicist. He published his paper in Science in 1964, where he mused about some patterns that he was observing in the scientific community. And one of the things that he noticed was that molecular biology and particle physics were just killing it. Just the progress they were making was like on a daily basis. It's amazing discoveries. Other branches of science, maybe not so much. So he published this very provocative thesis where he said, you know, these two fields of science are doing really well, and they're doing really well because of the culture of how they do science. And he identified something he calls strong inference, which is something that's sort of in the DNA of particle physics and molecular biology at the time. So he identified this sort of algorithm for what these people were doing to so powerfully make discoveries. And I'll just run through it very quickly. The first is, you know, when you have a problem you're trying to solve, come up with some alternative explanations. Devise a crucial experiment that will allow you to exclude some of those possibilities, and then carry out the experiment to get a clean result. This is not rocket science. This is not something that is surprising to anyone, especially in this room. And of course, you can recycle this procedure. But Platt presented this in a way that sort of reminded, especially biologists, that this is the essence of science and why science is so powerful, that you really can spend your entire career doing experiments and not making progress. So, you know, I'm sure what the philosophers in the room are thinking is, you know, isn't that just Popper's sort of falsifiability hypothesis? And the answer is yes. But this was a message that biologists needed to hear at this time. Okay, so what did I actually take away from reading Platt at the time? And this one was a really important thing to learn as a first-year PhD student. It occurred to me that, you know, I could spend several years spinning my wheels and not making any progress. So Platt puts it this way: we speak piously of taking measurements and making small studies that will, quote, add another brick to the temple of science, most such bricks just lie around the brickyard. Okay, so I definitely wanted to avoid making bricks that weren't going to end up in any structures. The other really important concept that he introduced to me was this idea of multiple working hypotheses. And this was not his idea. It actually came from someone named Chamberlain, who was also like Platt at the University of Chicago. He was a geologist. And I'll read you a quote from Chamberlain that actually appears in Platt's paper. The moment one has offered an original explanation for a phenomenon which seems satisfactory, that moment affection for his intellectual child springs into existence. To avoid this grave danger, the method of multiple working hypotheses is urged. Kelvin's talk a couple days ago got me thinking about this, because I think what Kelvin was suggesting in these adversarial collaborations is something that Chamberlain was suggesting we do sort of internally. So have an adversarial collaboration in our own brain where we entertain multiple hypotheses that are competing at the same time. So here's what Platt had to say about Chamberlain's idea. In fact, when there are multiple hypotheses which are not any one's personal property, and when there are crucial experiments to test them, the daily life in the laboratory takes on an interest and excitement it never had. And the students can hardly wait to get to work to see how the detective story will come out. So this was a sort of thrilling view of science that I really liked. And I can say that after adopting Platt's methods and his advice, the daily life in the lab did change for me in this way. Okay, and then, of course, falsification is the essence of what Platt is arguing with strong inference. And of course, this is what Popper was saying as well. What Platt does on this point, I thought was really powerful. He provides two questions that anyone can ask at any time to sort of get to the heart of the matter. One of those is, if you have a hypothesis, or if you're talking to someone who has a hypothesis, you just ask this very simple question. What experiment could disprove your hypothesis? Very simple. How often do we actually do it? And then another way of saying this is, if you have an experiment, what hypothesis is your experiment designed to disprove? And if you can't answer those questions, then maybe you need to think a bit more before you spend time at the lab bench. So in 2014, this was the 50th anniversary of Platt, I was inspired to write an homage to strong inference. It was published in the Journal of Experimental Biology. And, it was fine. A lot of people sent me polite emails saying, I'd never heard about Platt, cool to hear about it. Or I too loved Platt and he really changed my life. We read your paper in my lab meeting and it was super interesting. So that was nice, and sort of, helped me appreciate Platt even more. In 2018, shortly after I arrived at Chapman, I got an email from a guy named Barry Sharpless, who is a chemist at the Scripps Institute down in La Jolla. And this was an intriguing email to me. I hadn't gotten any emails from non-biologists before, and Sharpless is a chemist. And I'll read you a little bit about it. For more than 50 years, my two-word MO has been kill it. For the almost 50 years of my academic career, kill it has been my deflating response to excited group members who bring me promising but premature by my standard results. He talks about Descartes, so the philosophers will like that. He mentions Chamberlain's multiple working hypotheses. And of course, he talks about Platt and strong inference. And he invited me to come down and talk to his group about plaque. I was intrigued, so I emailed him back quickly and said, I'd love to come down and here are some dates. And then I Googled him and realized that he's a Nobel Prize winner, which was a sort of surreal thing to know I was going to go down and give a talk on scientific method to a Nobel laureate. So he won in 2001. So that was surreal to say the least, but I had a great day. Spent the whole day with him in his lab, and it was really enlightening. Then in 2022, an interesting thing happened. Barry Sharpless won a second Nobel Prize for a completely different thing that he had worked on. So I know what you're thinking. How much credit should I take? A second Nobel. And unfortunately, this was work he had done before I went down and gave my lecture. But, if I had any doubt that this method was a powerful method that could yield results, he's one of 5 people who's won 2 Nobel Prizes. And that includes, Linus Pauling and Marie Curie. Okay, what I want to do next is sort of unpack Platt's algorithm a little bit more, because it's really kind of cryptic. I tried teaching this to my students, and it's really vague. And so that's forced me to sort of unpack it for my students. So here it is again, devise alternative explanations, devise a crucial experiment, carry out the experiment. So let's think about that first one. These explanations, these are what we would call hypotheses. Okay. The second one, possible outcomes, that's pointing at something we would call predictions. Okay, so let's think about those two things a bit more. So here's that first part of the scientific method algorithm, part that I said has been ignored too much by me and others over the years. So let's think about questions for a second, because I think we can say some interesting things about the kinds of questions that scientists try to answer. Actually, Mike Ibba, and at the very beginning of this conference on Tuesday, said something very provocative, which was, the question at hand is, what is the question? Okay, so there's too many questions that we could possibly ask. So we need to be clever about which questions we ask. So let's come back to this, this misconception that I labored under for a while, where I thought, the best questions in science are the ones where you can say, what's the effect of this on that? And in retrospect, that really was something of convenience, because when you have data like that, it's easy to analyze them. You can do stats, and you can get a p-value, and maybe you can publish and be rewarded for that. Okay. But I really think this is a terrible thing for students to be taught. And what I think is, what I've come to realize is that the scientific method in its most powerful form that we all know and love was devised as a way to answer open-ended questions. Not these really constrained questions where you already know what variable A is and what variable B is. Okay, open-ended questions where you have no idea what the answer might be or how to even start to answer it. Okay, so what do I mean by open-ended questions? Let me give you some examples from my field of biology of some questions that, when they were first articulated, seemed like they would never be answered. And now we have really good answers too. all of these. Okay, what is the cause of disease? And I don't say infectious disease there because that's kind of the answer to this question. But if the observation is people dropping dead around you who were healthy 3 days ago, how do we understand that? That was a really important question that probably for a long time seemed like we would never have an answer to. And, we have the germ theory of disease now. Another question that people have provided answers to for millennia, unfortunately, all of them were wrong. We now have an answer to. Okay, where did life on Earth come from? Huge question, seems unanswerable. We answered it using science. Why do children tend to look like their parents? This is something people have noticed for millennia again. For a long time, it probably seemed like we would never know. And now we know, we have genetics. What's the molecular basis of that phenomenon that I just mentioned? Seemed like a really hard problem. We now know it's DNA. Once you know about DNA, well, how is that information translated and actually read within cells? Another really hard question. We know a lot about the answer now. I would argue that not only are these constrained questions sometimes, problematic and just way too constrained, but they actually can cause confusion if you try to apply the scientific method to answering them. So I want to just run you through a quick example of that. So I'm not going to talk too much about my work, but I'm a biologist. I work on biomaterials. So these are materials made by organisms. The most interesting biomaterial we work on is hagfish slime, which is what you're seeing in this photo here. So this eel-like critter, just a few seconds before, was happily swimming in this aquarium. And Atsuko, my technician, reached in and sort of grabbed the hagfish. And this is what happened. Okay, so they have this incredible defense mechanism where they can produce liters of slime in a fraction of a second. Okay, so that's one of the phenomena that I've studied in my lab. One of the sort of keys to the bizarre properties of the slime is the presence of these spider silk-like fibers that reinforce the slime. So you might be surprised to see that this slime is sort of supporting its own weight in air, which is not usually how slime works. That's because of these really strong, really tough microscopic fibers that reinforce it. The origin of those is these structures built within very special cells called gland thread cells that manufacture this 15-centimeter long fiber within their cytoplasm. And those things unravel when the hagfish releases the slime into seawater, and they end up reinforcing the slime. If you collect some of those skeins, as we call them, they look like little balls of wool, and transfer them into seawater, this is what they do. They spontaneously unravel. Okay, so here's a very specific phenomenon that might be puzzling, and it might cause us to ask some questions that we might want to answer using the scientific method. So let's do it the way that I don't recommend, which is let's ask a very constrained question. Okay, what is the effect of temperature on the unraveling kinetics of thread skeins from hagfish slime exudate? Seems like a very scientific question, right? It has technical words, temperature, kinetics, exudate, right? It sounds very scientific. But let's run it through the algorithm. Okay, so we've got a question. We're going to we're going to pose an answer to that question in the form of a hypothesis. We're going to hypothesize that the kinetics will be faster as temperature goes up. Okay, the next step in the scientific method is predicting. But our hypothesis is already a prediction. So what's going on here? We're kind of confused, okay? And I would argue that the way out of this confusion is to not even use the scientific method to answer such a constrained question. The experiment that you should do is sort of built into the question already. What is the effect of temperature on unraveling speed? Just go ahead and vary the temperature and measure the unraveling speed. Okay, you don't need to hypothesize all these different contingencies and predict, and you're just going to confuse yourself. If you want the answer to that question, go and answer it. Okay, the scientific method is there for us when we have no idea what the next step is. Okay, that took a long time for me to figure out, and hopefully it's not a totally obvious thing to everyone in this room. So let's see if we can ask some open-ended questions about this phenomenon. So one thing we might ask is, well, what's going on? What's driving that unraveling process? What's the biophysical mechanism? Is it obvious what the experiment is there? No. Is it obvious what potential answers are? No, we have to put on our thinking caps and come up with some creative ideas. One reasonable idea is that there's like a swelling, osmotic swelling that happens that drives the unraveling. People have suggested that. Another possibility, a little more elaborate, turns out to be the true explanation, is that there's an adhesive on the outside of those skeins that holds it together. And there's actually bending energy in the thread that gets released when that glue softens and dissolves away in seawater. Okay, so this is a case where using the scientific method, hypothesizing and then predicting actually would help a lot. There's another open-ended question. What is the mechanism by which thread skeins unravel to 1,000 times their original length in under 400 milliseconds, which is how fast it happens, when a hagfish makes slime in the wild? Again, no idea what the experiment is that's going to tell us that the answer to that question. What do? We put out some hypotheses and then evaluate them. So maybe turbulence is really important for pulling the skeins apart. Maybe the mucus part of the slime is important. Maybe it swells and loads the skeins in tension. Okay, maybe it's something else we haven't thought of yet. Okay, so these are open-ended questions. One more that I'll throw out there, which is one that keeps me up at night, which is, how in the world do the cells that make these structures make these structures? Okay, this is something that's built in the cytoplasm of this cell. It takes them about a month to do it. And again, it's 150 millimeters long in a microscopic cell. Okay, so they're basically making spider silk within individual cells. Okay, so that's what I wanted to say about questions. What about hypotheses? I mentioned before this idea that is crammed into our heads in school, that a hypothesis is an educated guess. And the terrible thing about that is that it implies that what we do in science is we design an experiment and then we just guess about how it might turn out. And that is not how we do science. Okay, so I spend a lot of time in my classes trying to delete this concept from my students' brains. So instead, I try to replace it with this concept of hypotheses, and that is that hypotheses are explanations that represent potentially satisfying answers to a question at hand, an open-ended, difficult question. So one of the things we've learned over the years about evaluating hypotheses, when we do this with students and we're at the whiteboard and we're generating hypotheses and we're trying to figure out which ones are good and which ones we should leave behind. The first thing we ask is, is that a satisfying explanation to the thing that we're trying to explain? It also needs to be free of contradictions internally. It should be, it doesn't have to be, but it's always good if it's consistent with existing knowledge. Maybe you're on the verge of a really big discovery, which is going to overturn existing knowledge. So you don't want to be too strict about that one. But we can use existing knowledge to sort of shape what we think is reasonable or not. And this is another useful one, which is that when you're doing predictions, does the hypothesis predict the original observation that you're trying to explain? If it doesn't, then it's not a hypothesis worth considering. Okay, so the title of my talk mentions predictions, so I want to spend most of the rest of the talk talking about predictions. And so here's that beginning of the algorithm that we've ignored for too long. We've talked about questions and hypotheses. Where do predictions fit in? Well, right in between hypothesizing and testing. Okay, so let's talk about that for a second. So hypotheses, as I've defined them, are abstract explanations of how things work. Experiments are things we do in the real world with our hands. And, those two things don't talk to each other very well. And that's where predictions come in. Okay, so predictions are a bridge between abstract explanations and experiments we can actually do in the real world. Okay, hypotheses, because they're explanations, they're usually kind of abstract, and it's not obvious how to test them. Okay, so predictions get us there. They help us get to something we can actually do in the real world. Okay, this is something that everyone in this room already knows, but students don't know this. For predictions to function as a way of disproving a hypothesis, they must flow logically from the hypothesis. Okay, the error that students make is they think, oh, I'm going to predict how the experiment is going to turn out. No, it's not you predicting, it's the hypothesis predicting. You have to put yourself in the mindset of the hypothesis being true, and then think about how the world would look under a given set of circumstances. That's what it means for the hypothesis to predict. You can predict whatever the heck you want. It's not going to have any bearing on, how the whole thing is analyzed at the end. So I taught a field course with a former student, Andy Turco, for many years. And we spent a lot of time with students at the whiteboard doing the kinds of stuff that I've been talking about for the last few minutes. And we sort of stumbled on this concept of critical predictions and persuasive predictions. And I'm really interested to hear what especially the philosophers think about this idea. So I want to unpack that for you in the next few minutes. So when we're at the whiteboard and the students come up with predictions, it would be nice to have some criteria for figuring out, what are good predictions that we should actually test? How are we going to spend our precious time in this course in the lab? So the first one's kind of easy, and this relates to Popper and strong inference, and it has to do with falsifiability. And it has to do with this question. So if the prediction made by the hypothesis is found to be false, would it force you to reject the hypothesis? Okay. If the answer to that is yes, we would call this a critical prediction. Okay. It's a really good prediction because it has the ability to wipe out one of your hypotheses, and that's progress. Okay. You're that much closer to knowing the answer. But what about the other scenario? What if you find the prediction to be true? Is there anything to be learned from that? And what Andy and I argue in this paper is that yes, there is. So if the prediction is found to be true, would it help convince a skeptic that the hypothesis has some merit? And if the answer to that is yes, we consider that a persuasive prediction. Okay, so I realize this is quite abstract. So the best way to lead you through this is with an example. This is actually an example that came up teaching this field course. It might even be the example that led us to the idea of persuasive predictions. So we were out doing some work in the field. We were actually looking at a tide pool. One of the students noticed these tiny little organisms called springtails floating on the surface of the tide pool. So springtails are arthropods. They're sort of insect adjacent. They're not insects, they're hexapods. And these are really weird springtails in that they live on the surface of tide pools in a marine environment. And what the students got interested in by looking at these little guys is the fact that they all seem to clump together. Okay, so that was an observation that they thought was interesting. This is not an important, phenomenon or question, but the question is, why do intertidal springtails form rafts? Okay, but for the point of explicating these two types of predictions, it's a useful example. So the first hypothesis that the students came up with was, oh, these are mating aggregations, right? They really look like active structures, and it's males and female springtails getting busy. Now, we did our usual thing and we said, okay, if that's your hypothesis, what does it predict? And one of the students raised their hand and said, well, if that hypothesis is true, then this species must be a sexually reproducing species. And we thought, okay, let's ask our quality control questions about that prediction. So the first question is about criticality. So if you found that this species was not sexually reproducing, would that force you to reject the hypothesis? And everyone sort of thought about it for a second, and there was a lot of nods. And people said, yeah, if they're not sexually reproducing, then they can't be mating aggregations. Okay, so check, this is a critical prediction. Okay, but let's turn it around and ask, what would happen if we got the other result? What if we checked and we found, oh, they are a sexually reproducing species of springtail? It really wouldn't move the ball down the field very much for our hypothesis, right? Because the vast majority of species in the world are sexually reproducing. Okay, so this is an example of a prediction that is critical, but not at all persuasive. So is it worth testing? So I'm really interested to hear what the philosophers have to say about this concept of persuasive prediction and whether it exists or not. But one important question, I think, is, can a prediction be completely persuasive? And I think the answer is simply no. because, we never prove hypotheses. We always leave the door open that there's a better explanation. So there should never be a moment where a skeptic says, I am 100% persuaded. So this makes the concept a little fuzzy because we can never achieve, full persuasiveness. Does that mean it doesn't exist? I don't know. Just to carry this a little bit further, are there other viable hypotheses we could think about? A reasonable one is the Cheerios hypothesis, which says that, it's not about mating aggregations. These things they can't help it. They're little objects floating on water, and because of surface tension, they tend to stick to each other like Cheerios in a bowl of milk. Okay. Interesting hypothesis, might explain everything. Let's take this apart. So let's think about the Cheerios effect. Well, one prediction that the students came up with is, well, if that's true, then if we add a little bit of surfactant to the tide pool, maybe a drop of soap. If it's true, then the effect should go away. Okay. Well, let's think about if that's a critical prediction and a persuasive prediction. Okay, so the prediction is adding surfactant will eliminate clumping. So let's think about that. If we add soap and they don't drift apart, would that force us to reject the Cheerios hypothesis? And we always let the students think like this for many pauses, because it takes a while to think it through, right? I think the answer is no, it wouldn't force you because you don't know enough about what's going on. Did you add enough soap? Did you add the right kind? Did you disrupt the surface tension enough to really abolish this effect? Okay, so here's a case where it's not a critical prediction. Is it persuasive? What if you add soap and they all just drift apart? Would that persuade you that the hypothesis has some merit? I'm seeing some head bobs. Okay, so I think so. I think there's some persuasiveness there. But of course, so I lost a slide. Okay, so that's an example of a prediction that is not necessarily critical, but could be quite persuasive if you got a certain result. Okay, so what's the point of doing this? Why ask ourselves about criticality and persuasiveness? This is not a magic bullet for helping you find the perfect experiment, but it does help you think about, what exactly are you going to learn if you do experiment A versus experiment B? And what are you going to learn if the experiment goes this way or it goes that way? And it's always good to think those things through before you put in the work. Okay. And what we found is that asking these questions in an iterative way helps us improve our experiments hugely. Okay, so I've given lots of sort of practical advice about how to navigate some of these aspects of the scientific method, but what can we do with our students to help them learn this better and avoid the mistakes that I sort of fell into over the years? So one of the things I do in my lab is students actually do, they use strong inference in the lab. So they come up with questions, they make observations, they hypothesize, they predict, and then they actually do the experiments in the lab. I mentioned this field course that I teach. This is a really perfect place to teach scientific method and strong inference. We spend a lot of time in the field, a lot of time at the whiteboard, students designing experiments, doing experiments. But you can't do that with every student. And sometimes, you know, I've been invited to elementary schools to talk about scientific method. And, you know, we can't do experiments in the hour that I have with students. So I'm going to show you a little video of something that I've used over the years to teach some of the concepts that I've been talking about for the last little bit. So it doesn't have to be this video. It can be any sort of puzzling observation that people are going to be surprised by. Okay, so this is something that somebody found on the internet and sent it to me. It's a Chinese variety show. Okay. So I heard a lot of kind of laughing and some raised eyebrows. So that's the point of this, is that it's puzzling, right? You don't need to know much about the world to be puzzled by this video, okay? You could do, you know, a similar demo with like real data and ask students to work through that. But if you're working with fifth graders, you want to use something that they're just going to latch onto and be puzzled by and immediately start asking questions about. So if I had another hour, we could actually go through some hypotheses that you have about what's going on here. I don't think we have time. I'd much rather use that time for discussion. But that's just an example of the kind of thing that you can present to students to get them puzzled and engaging with the scientific method. Okay, so a few take-home messages to end, and then we'll take some questions. So these are probably not things that people in this room are terribly surprised by, but these are the kinds of things that I like to leave my students with. So the first one is open-ended questions. Do not shy away from open-ended questions, even if they seem completely unanswerable. So Jack Horner, who's sitting right here, is probably the person who I know who asks questions that other people think are just unanswerable. And he's done that over and over again and found answers to these open-ended questions that on the surface seem just completely impossible. Don't just strive for publishable data with significant p-values, but strive for explanations. That's going to be much more rewarding. When you're generating hypotheses, don't get attached to one hypothesis. Generate multiple working hypotheses and then pit them against each other, figure out where they diverge, come up with predictions, ask if they're critical and or persuasive. If the answer is not so much, then try to make them more so. I should also mention that critical and persuasive are not mutually exclusive. In fact, you can have one prediction can be both critical and highly persuasive at the same time. And then this last point, I think is really important. So if you come up with a list of hypotheses, you do your careful experiments, and you disprove all of them, don't despair. This has happened to us many times. You have to think about that as progress. If your hypotheses were reasonable and you disprove them, what you've shown is that reality is much less reasonable than you initially thought. It's more interesting, it's weirder. You're gonna have to come up with a weirder explanation than you initially thought. Okay, I'm gonna end there. I'm gonna acknowledge some people who helped me on this journey. I want to especially thank John Gosline, who was my PhD advisor, and he sort of embodied the strong inference approach in how he thought and how he talked and how he wrote. And it really was kind of amazing to learn how to think like a scientist from him. Okay, I will end it there.
So I see a question from Lou.
Yeah, as I remember at class, he was making an argument, well, at rapid progress, right? That this leads to more rapid progress in science.
Yeah.
Do you think that's really true? I mean, what you talked about was more like it's more satisfying science. Does that necessarily translate into rapidity of progress?
I think it probably does. I mean, I've flirted with the idea of trying to figure out, if people who use strong inference, and I don't know how you figure that out, but I guess you figure it out from their papers.
It's an open-ended question.
If they do, I mean, it's the bibliometrics stuff that gets really hairy. But you might be able to test that if you're using words like, if then prediction hypothesis, do you end up making a bigger impact and making more progress? I'm not the person to probably answer that, but
I think that's an interesting.
Yeah.
There was Marco.
Yes, thank you very much for this splendid lesson of philosophy of science. I'm not a biologist and I'm not an experimentalist, but I spent part of my young life to study philosophy of science. So I have some reaction to you. The first one is possibly the only one that I will mention. You mentioned Popper, and I think that behind what you say, there are much. But there's something in Popper that play a crucial role that seems to me a little bit obscure. It is the notion of background knowledge. The fact that in an experiment or an hypothesis is never alone, but only relative to background knowledge that play an essential role in deciding how to evaluate the outcome of an experiment. So what I would context in our description is this idea that the dynamic of science is something as a sort of, so to say, an hypothesis and an experiment. And let's try to see how to formulate well the hypothesis in order to conceive the good experiment. But I think, I don't know, I'm not an experimentalist. Perhaps your answer is no. In my experience, things does not work this way. But it would mean that your experience deny my idea. That perhaps experiments are also done in order to imagine experiments. It is not only to evaluate, to exclude, or to confirm, or to be persuaded, as you say, about an hypothesis, but also to imagine it, to identify the category that we can use to describe the phenomenon. No, I'm not a biologist, but if I take this and I see that, I can read it in different way. I can read it as a body that go up to the bottom, or I can see it as something that has a certain speed and a certain trajectory and goes up to the center of the Earth. There are two totally completely different descriptions, and the hypothesis depend on the categories that I use. So it seems to me that the dialectic between hypothesis and experiment, but you do not deny in a certain sense because is there implicitly. But it seems to me that it would deserve a more explicit, more explicitation.
Yeah, and I think that's right. And I think what this ignores is the role of sort of exploratory science, which is not hypothesis driven, where you're just measuring some stuff. right? Like the Human Genome Project was not strong inference. It was just measuring a whole bunch of base pairs and reading them out. Now, once you make that observation, then there's interesting puzzles that you might want to try to crack open. And that's where you might need this.
So neighbor of Marco.
Somebody else have a question?
No, I know, I have noticed two other questions, but there is an order.
Can I also make a remark? Can you stay?
Oh, yeah.
Okay.
Okay, so quickly, a quick follow-up on the first question related to Platt. It's gonna matter what counts as progress to be able to answer that question. And your last slide highlighted that the disproving of all of one's hypotheses should not cause despair. Right? Well, in part, that would be because it's a kind of progress, but maybe not the kind of progress that everybody envisions. So that's, I think, an interesting thing to raise is what kind of progress do you expect? Maybe there are ways in which rates of progress are higher for strong inference patterns, but maybe not the progress that one would necessarily expect.
Yeah, and I think what you're assuming is that a negative result can't be published. And, you know, I think we need to rethink that, because if it's a reasonable hypothesis and you disprove it, that's an important result and should be published.
So the other question that I had is related to how you think about why critical and persuasive are relevant categories for assessing hypotheses and predictions, or predictions in particular, in cases when you presented it. It seems like what you want is not just that you can call it critical or persuasive, but that you can give an account of why it's critical or why it's persuasive. And so what's really at the root of that is the ability to be explicit about the basis of critical or the basis of persuasive. And that becomes especially crucial in contexts where there might be different evaluative standards, say across disciplines, where what might be persuasive to one discipline is not persuasive to another discipline. And part of the way of getting to the bottom of that is saying, well, why would I think it is or isn't persuasive? But I take it that's probably something you would.
I mean, that's part of the really fruitful discussions that come up when you ask the question. But again, I think, you know, you can't define it as because you're never fully persuaded. It's inherently kind of a fuzzy concept.
But you do want to be able to say why I would be persuaded.
I have four more
Yeah.
questions, so.
Okay.
First speaker of the day.
Thank you. So I would think that it would depend at some level on the position of development of a given field, in other words, where they are in their development. Seems to me there was a time, like you said, for exploration, and for describing. Because your hypotheses are going to be well served by knowing what you're doing.
Yeah.
And so I would think that there's a strong component of where you are in the development of subdiscipline.
Yeah, that's a really good point. I mean, I'm not advocating that this is how all science should be done. What I'm saying is, if you have an open-ended question, you know, this is a great way to sort of enter it. If your question is exploratory, if your question is how many microbes are there on the planet, you know, it doesn't help to say, I hypothesize, you know, there's 2.5 million, right? Makes no sense. You just go and you do it.
Andrew Jordan.
Yeah, so thanks so much for the nice talk. I really enjoyed hearing about these spools of slime producing things. It's amazing. Nature always surprises us. One word that stuck out in your talk that jumped out at me was the word satisfying, adjective satisfying about your hypothesis. And I wanted to attack that idea a little bit, because there are many areas of science where our best scientific descriptions are not only unsatisfying to many people, but indeed downright disturbing There are whole fields of physics where people are raging against this field because they hate what it is teaching us about nature. So can you, from a philosophical point of view, defend this notion of satisfactoriness or satisfying as a defensible adjective for your hypothesis?
I think it relates to the last point in that slide, which is that, does that hypothesis predict the puzzle that you're trying to explain? So, and I would just leave it at that. So that's my definition of satisfying is, does it predict it? It's not an aesthetic thing of, do I like this idea or does it fit with my worldview? Something like that. I think we can really narrow it down to, does it predict in a way that explains?
Eric?
So mine's a follow-up on, I think, the first question about how we measure progress, but I also wanted to make it more precise. Platt held up particle physics as the place where this happened.
Yeah.
All his rapid results, but arguably it went into stagnation not long after this paper was published. And in the philosophy of science literature, I think there's a broad consensus that scientists throw the kitchen sink at problems, and then some methods work and others don't. That's not my own view, but this is sort of consensus. But I think here you have a hard prediction, as it were. And looks like this method didn't work so well in particle physics after it was announced. So maybe they want. I mean, I think there are other things happened there too, but it seems kind of ironic that this is your. I like the Hutto paper, by the way, very much, but I think that method is different.
It is different, yeah. That's a good point, and people have criticized Platt for, not recognizing the sort of domain-specific nature of the problems that molecular biology was working on at the time and particle physics working on at the time. And sometimes there's just a kind of fortuitous nature of, what people are given from their predecessors that leads to a really fruitful time.
And then a gold design sometimes.
Michael?
This is really neat. Your discussion of critical and persuasive predictions made me think fitting them into a framework of Bayesian inference. It seems like in both cases you're saying I want to choose an experiment that will maximize the change in my posterior belief in the hypotheses.
Yeah,
Is that
I don't know enough about Bayesian stuff, but I've heard people talk about credences and belief in credences that I think that may be what we're talking about with how persuasive something is how much does it change your credence? Does that make sense? Yeah. Is that a word that they use in Bayesian?
Yes.
Yes. Okay.
A lot.
Do we have another question?
There's one on Zoom.
One on Zoom.
Okay.
Mina, so you want to unmute yourself and ask a question?
Yeah, thanks, Doug. It was great to hear how science works in other fields. I had a very brief question. You show the diagram from question to hypothesize, predict, experiment. Can you think, I can think one in physics, where in fact we'll start from experiment and go through the cycle that you mentioned. For example, the Michelson-Morley experiment had some information, and then what led to was the question of hypothesize, predict, and look back to the experiments and verification of the hypothesis.
Yeah, I mean that's a good point. That it's not a linear process, and nobody starts at the beginning and ends at the end. So what I'm saying is that if you have something that's a puzzle that needs explaining, then that's the observation at the top. And you can sort of run through the algorithm to try and find an explanation for that. That's not to say that you can't just go out and do experiments. And sometimes those experiments might lead to really interesting new puzzles that are going to raise new questions that need answers.
Thank you.
Margaret again.
Yeah, thanks very much, Doug. Are you gonna tell us what those fish were doing?
Yeah. Oh, okay, how much time do we have?
9 minutes.
9 minutes, okay. Let's hear some hypotheses from the crowd.
They're
Yeah,
remote controlled,
okay, remote, what do you mean by remote control?
they're not real fish.
Not real fish, okay, so robot fish.
Magnets.
Magnets.
Magnets, say more. What do you mean by magnet?
Well, I mean, it looked like a magic trick where there's some automaton under the tank that's running magnets and the fish are following magnets because they have.
So there's a magnet in each fish,
Yeah,
Mhm.
something like
an elaborate mechanism under.
Somebody's using lights on them.
Okay, so we've got magnets, we've got robo fish, we've got lights dazzling the fish. Sound, okay, so I think they want us to think that it's the sound, there's a whistle, he's talking to them, waving his hands. Okay, so we've got 4 hypotheses now.
Video is fake.
Okay, it's a deep fake. When I do this with 5th graders, and I've done it several times with 5th graders, they fill the whiteboard in about 5 minutes with insane hypotheses. Eventually they get to something reasonable, but they're amazingly creative. But we have 5 hypotheses now.
Can I tell a nice story? In Italy, when we that story we take with carabinieri, I don't know, the carabinieri, I don't know what is in English, the sort of policeman, but not exactly the policeman, part of policeman of the army. And there is a nice story, it is the following, this person like this one, that was explaining to a red fish how to move. Finger here and say, go up, and the fish go up, go down, left, fantastic. So came back, I am not only able to command myself, okay, I'm only able to make you command yourself. So Alex, came here, please, put it, command, down, right, left, said, then Bob, came here, again, and finally the carabiniere, so came here, command.
Okay. Let's talk about the magnet hypothesis for a second. Okay, what does the magnet hypothesis predict we should see if we could do some experiments or even analyze the video?
That there's a mechanism underneath, and that each fish has a magnet, and those mechanisms are moving in different directions, different areas.
Okay, so one prediction is that if you were to cut open each fish, you would find a magnet inside it. Yeah, that's true. You could just stick a magnet up to it. Is that a critical prediction? If you didn't find a magnet inside, would you be forced to reject that hypothesis?
No. I don't know more about.
No?
No.
Why not? Why no?
It could have magnetic glands.
Yeah,
Maybe
some animals locate by magnetism.
Okay, but if we go out on a limb with this hypothesis and say it's an actual magnet that somebody put there.
Yeah.
Okay, and this is a good thing to do, is to go out on a limb with your hypothesis and like really make it as explicit as possible, because it makes it easier to eliminate it when that's the case. Is there anything we could do analyzing the video to test this idea that they're actually being controlled by magnets and not by movements of their fins, which is how a fish normally moves?
And the way they cross each other's paths would, I mean, if they're all controlled by magnets, the magnetic fields are sort of interfering with each other in some configurations, you wouldn't be able to get all possible movements, I would think.
Okay, so you're saying that we might be able to analyze the movements and rule out magnets if we show that certain ones are not possible with magnets. Interesting.
So what do we know about fish locomotion? So if it moves backwards without using its fins, then that's pretty.
Okay, so we know a lot about fish locomotion. There's people who do fluid dynamics of fish swimming. they could actually analyze the movements of the fins and tell you how that fish should be moving. If you can show that there's a disconnect between that and how the fish is actually moving, then that's probably very persuasive evidence that the magnet hypothesis is right. I don't know the answer. I think it's magnets. But
I don't know the answer.
I don't know the answer. But
Don't sleep tonight.
my students sometimes want to murder me when I do this, and I tell them I don't know the answer. They're like, what?
Is it the Cheerio effect?
Oh, I think it's the Cheerio effect. Yeah.
So after this confession, it's time to thank again.